So these are some of the additional online supporting resources that we have for students, and this particular presentation focuses on good practice guidelines for the production of supporting figures and tables that you might report, for example, in a dissertation or a classwork report or maybe a lab practical. The idea here is to help you to understand the fundamental principles of how to present your results clearly and the things that will make your tutors and the markers of your work smile. So in this next slide we're looking at the production of figures, and what I've done here is show you good practice on the left and poor practice on the right. So let's first of all look at what constitutes good practice. So we're looking here at the figure on the left. What we really want to see is a clearly drawn figure with nice clear fonts on the X and the Y axes. We want to see the appropriately used techniques. In this case we're using a histogram with error bars shown, and also some indication of where significant differences occur between different categories, in this case related to depth ranges. It's important also that we know what these figures are showing us. So we know very clearly that the categories we're looking at are depth range, but importantly we're also told what the units are. So in this case depth range measured in meters. On the y-axis we're interested in knowing what our response variables are. So we have two graphs here, one related to mean bivalve biomass, again with units shown, so that's in grams, and mean bivalve density, so that's number of organisms per 0.1 meters squared. Straight away you've spotted a slight error here in that the top graph should report biomass as grams per meter squared, so there's an immediate improvement that could be made. Now a figure should be able to stand on its own. In other words, if you take it out of the context of your report, you should just be able to read the legend, view the images in the figure and understand what the figure is trying to tell you. So first of all we have a label, so this is figure 2, and then straight away it's telling us what we're looking at. So we're looking at mean, and then we're told what the error bars equate to. So we're looking at the mean plus or minus the 95% confidence interval, bivalve biomass, grams per 0.1 meters squared. So here we are told what the standardization and units are, and mean bivalve density per 0.1 meters squared again. For each depth group, or you could use the word category here, in meters. It might seem repetitive, but it's important to be precise. For what? For infaunal samples collected from where? From Red Wharf Bay, which is a location in Wales in the UK. Then of course we've used some symbols in our figure. We've used asterisks. So what do those asterisks denote? So here we explain that. So we say asterisk denotes a statistically significant difference between the two means identified. So obviously the ones which are connected by the lines. And the test is computed using the Student-Newman-Cool's post hoc multiple pairwise test. So basically we have all the information within this legend to understand what it is that we're looking at, where the data was collected from, the type of data it is, and also how any tests that are displayed in the figure are actually computed. Now if we look at the example on the right hand side, so this is poor practice, well straight away you can see the figure legend has far less text in it. So clearly that's an indication that there simply isn't enough information. So the text legend lacks sufficient information for us to understand what this figure is telling us in sufficient detail. Also, if we look at the figure itself, well, to the large extent the figure is identical. But if we look at the axis labelling, we can see that the axis labelling is not very informative. So we're not given units, for example, and also the axis labels are in the wrong orientation. In addition, the font for the axis labels could be far larger to make it a lot clearer and easier to read. So hopefully that's informative. Now let's move on and look at tables. So tables maybe not as exciting to look at as a figure, but a very efficient and informative way of presenting a lot of data. And again, the same principles apply. Now, very often in tables we use abbreviations, and it's essential that whenever we use an abbreviation, we explain what that abbreviation means. It's also essential that if we foreshorten terms, we also explain what their full meaning is. Now let's look at the best practice example, which is the one on the left. Again, we can see the legend text is lengthy, and it's lengthy for a reason, because it's a complicated table. So it's labeled table 2, and this is a table of the mean abundances, diversity indices and sediment composition. So again, we're told that the values displayed are means, plus or minus 95% confidence intervals. That could be foreshortened to capital C, capital I. Calculated for benthic informal communities, so that's what is the data derived from. Sampled at each depth group, and the results of the analysis using a one-way analysis of variance, brackets ANOVAR for short, or a Kruskal-Wallis test. So in other words, not only does this table display the mean values together with the error values, but also embedded within this table is the outcome of an analysis of variance or a Kruskal-Wallis test, together with the p-value for the result as well. So this is a highly efficient table. You might think it's a little dense, but there's everything in here that I actually need to understand what is going on. There are also some superscripts. They're quite important as well. So for example, some of these metrics, so the response variables, were transformed so that the data met the assumptions for the statistical test used. And that's very efficiently shown by using a superscript. And the superscripts are explained. So superscript 1 and superscript 2. When we look at the table itself, the text is relatively rich. So we're told we're looking at the total number of individuals per sample, the total number of species per sample. There's a nice heading that demarcates where we're showing the diversity indices for each of these samples. So P allows evenness, species richness, Shannon-Wiener, Simpson's index. And then for the analysis of the sediment parameters, and again a nice little heading saying sediment, and then percentage of fine silt or percentage of fine mud or percentage of sand. If we look at the poor practice table on the right hand side, well again quite clearly the legend text is very thin and simply lacks sufficient detail to enable us to understand what's in this table. In this particular case, the column headings are not well explained either. For example, they're just categorized as 1, 2, 3 and 4. So I know it's a depth range, but all I know is it's depth range 1, depth range 2, depth range 3 or depth range 4, but I've no idea to what that relates. I'm lacking some information for my variables, so I know I'm looking at individuals, abundance, maybe, but is this the mean? What are the plus or minuses? I don't know that either. And I'm given a p-value of some supposed statistical analysis, but I've no way of knowing what that statistical analysis was. Now this might be explained in the text, but actually this is a missed opportunity. A table presents you with a really nice opportunity to give a lot of information in a very efficient way, where it's all lodged together, either in the legend or the actual table itself. So this table on the right hand side is particularly uninformative. So hopefully these pointers will help you think about these things in the future, and maybe come to love your figures and tables just a little bit more, because at the end of the day they are your friends in telling your important story.